Sorry for the delay. Hopefully that goes back to. Okay. So hopefully I've resolved my problem with my computer crashing. I spent a lot of quality time with a nice young man from OTS on Friday. So please give me some feedback on the quality of the next recording because it's using QuickTime versus the other program I need to use. Okay, apparently the other program was sucking all the memory. It was weird. It was like we watched it and it was like tons of memory and then it would be like crash. So I don't know, something's wrong. Okay, lecture 8. So I did manage to salvage lecture 7, and that's up. Lecture 6 got destroyed somehow with a crash. So my apologies. Feel free to look at past year's lectures. It's generally the same stuff. You're just going to have to poke around. Okay, quick. Open recitation this week. That means that I'm trying something new. Usually I do these practice problems for the recitation before an exam, and everyone can kind of work through them together. But what I find is about 50% of the class is always very frustrated because they haven't had time to really start studying yet, and it's not useful. So it's open recitation, meaning I'm available between the hours of 1 and 5 today, and between the hours of 1 and about 4 on Thursday, so you can come and find me either in 217, so if you want to meet with your group and work through it like a normal recitation, you can do that. Or you can use that time to talk to me about things, or you can use that time to work on your picture of all pictures as you integrate all the different parts of the class so far into one picture. You can use that time however you want. You cannot come. It doesn't matter. If you're like, I need this time to study by myself in a dark closet somewhere, you can do that. I have no problems. You will not lose 10 points if you don't show up, but I am there if you would like me to be there. Okay. I've gotten a lot of emails like, wait a minute, so you don't have to come? I'm like, no, you don't. But you should. Exam 1 is next week. It's during your recitation time. Bring your laptop. If any of you guys have a laptop you find to be really reliable, can you just burn it just for kicks? Because I'm nervous that we're going to get too many people in the computer lab and have computer problems, meaning not enough computers. So if you can bring your laptop, you'll still use the computers in the lab, but if there's a problem, at least someone can say, hey, I have my laptop. Exam questions are due on Friday by 5 p.m. That means that if you would like to try your hand at getting up to 2 extra credit points for submitting a quality multiple choice exam question, then please do so. That is due by Friday, 5 p.m. By quality, I mean it cannot be a recall question. It can't be like the definition of RAM is. No, that's bad. All 4 options, there have to be 4 options, none can be all of the above, none can be none of the above, because those are cop-outs, only I'm allowed to do that. And they all have to be real possible answers, so one can't be, what do you mean cell biology? I thought this was physics. I don't know.
So each question that's accepted is 1 point?
Each question that's accepted, if I don't have to do anything to your question, you can get 2 points. Like if it's perfect and flawless. And every now and then I do get those. You get 2 points plus the value of the question when you see it on the exam and you hopefully know the answer. If it needs a little editing, you'll get 1 point. So there's your options.
You can submit 1 question.
You can submit more than 1, but please do not send me 20 of them. I don't usually have the time to go through about more than 2. So if you're like, these 2 are so great, I don't know which one's fine. But you only receive credit for 1. Yes?
Email them to you?
Email them to me, yeah. That's by 5 p.m. Friday. And then someone reminded me that I usually give an option for an oral exam. So for those of you, Usually people don't do that on the first exam because they're trying to feel out and see how the exams go. But if you're one of those people who's like, I can talk about this forever, but the minute I sit down in class, my brain goes blank and I don't know anything anymore. Feel free to talk to me on an oral exam. Usually what I do is I'll say, hey, tell me about this, and I'll keep questioning you until you can't answer anymore. And then kind of give you like, you know, A, B, C, D, F based on the percentage of that. Really a combination of the percentage of the things I'm looking for that you mentioned and the amount of prodding you need from me. Like, well, maybe if you think about this, then you're going to be losing points. But sometimes it works really well. I've had people get an A on an oral exam when they've been failing every single in-class exam. So, who knows? So, if you are interested in that, please email me this week. Yes?
I think I read in the syllabus, do you drop the lowest exam?
I dropped the lowest exam.
Okay.
Yeah.
For the optional oral exam, is it just one question that we just talk about relentlessly? Or
Yes, it's pretty much one question that can morph into, if you notice, there's really only one topic on this exam, right? Which is transporter proteins. So your one question might be, tell me about transporter proteins in the cell. And it's this open-ended thing, and you might say, I'm going to start with talking about circulatory pathway, and you'll start talking about, well, what about proteins? You know, it'll just be a conversation, and if you hit all the points and can talk purely about them, then there's no reason you shouldn't get an A, right? Yeah.
How many open-ended questions?
How many questions on the exam? Open-ended, you mean short answer? Between 2 and 4, depending on my mood. Yes. We're still meeting for lecture, yes, yes. So you will have to power through this when you're really wanting to be studying. Yes.
The exam is going to be from lectures 1 to lecture 8 or 9?
The exam will be through whatever we cover by the end of Thursday. Yes. Everything talked about on Thursday will be on the exam. Okay, ready? Are we good? Okay, so take 5 minutes to write. Okay, so this I kind of harped about for a while, so it's kind of a giveaway, but this will be a good way for you guys to test yourself to see if you understand this. And this was mostly inspired by the fact that I got some interesting questions over the weekend with some of with a quiz, and so I want to make sure everyone's understanding this. Proteins go to the lysosome for different reasons. And therefore, right, because there are different purposes to why a protein might go to the lysosome. And therefore, they take different pathways to get there, right? We spoke about 2 different pathways for getting to the lysosome. Discuss these 2 different pathways, providing examples of a type of protein, so a category, a major class of protein, providing examples of a type of protein that would take each pathway. Like this protein is likely to follow this pathway. This protein is likely to follow this pathway. Explain the mechanism of how each protein enters the pathway and be sure to include any mechanisms of regulation involved. So this is very kind of big and open-ended, but I want to see if you guys can differentiate. Okay, take 5 minutes. Okay, how's it going? Need more time? Okay, I'll give you guys another minute or two. Okay. So, how about Christina? Where's Christina? Ah. So, tell me how you approach this question.
I kind of did a little bit of an overview of like the anesthetic pathway and secretory pathway. Basically saying with like the anesthetic pathway that a protein is coming outside of the cell and traveling into the end of the cell to fuse with the lysosome. And then that this pathway is detonated due to acid hydrolysis. And then the secretory pathway would include the protein entering the ER and traveling through to the Golgi that goes to the lysosome.
Okay. So, Christina said she kind of did an overview of the endocytic and secretory pathway, with the endocytic pathway being the way a protein is going to come from outside of the cell through endocytosis and be degraded in the lysosome. And the secretory pathway, where you have a protein that goes through the ER and ends up going to the lysosome to work in the lysosome. Okay. Does anyone want to add to that? Did most people kind of get on that path? I see nodding. That's good. I saw a lot of furious writing, which is good. So what type of protein would be following the endocytic pathway? Yep. LDL receptor. Yep. Yeah, so receptors, anyone who wrote any sort of receptor or a ligand bound to receptor such as LDL, that would be correct. What kind of protein would be following the secretory pathway to end up in the lysosome? Acid hydrolysis. Good. Okay, so you guys are on the right path. And then when I said something about explain how they get into the pathway, we're not going to worry too much because I think you get that. How about any mechanisms of regulation? So what mechanism of regulation is used for a protein to be endocytosed? Yep.
Would it be like the pH is and the acid hydrolysis?
So okay, so you're thinking about activity of acid hydrolysis and the pH.
Yeah.
So that's what's going to determine whether a protein is going to be degraded once it's in the lysosome, but in terms of inducing endocytosis, something that's going to signal endocytosis. We spoke about it for 2 seconds. Yep. Clathrin mediated, but what regulates that? So we talked about monoubiquitination of receptors and that induces endocytosis. That's regulation. Something's got to say, hey, endocytosis, right? So that's ubiquitination. Monoubiquitination of your receptor. And mechanisms for proteins to make it to the lysosome through the secretory pathway. Any mechanism of regulation there? Mannose-6-phosphate, is that what you're going to say? Yeah, mannose-6-phosphate. Yeah, okay, so if you guys were kind of generally on that path, congratulations. And if that's kind of the depth that I'm looking for in these open-ended questions. Okay, let's move forward. Yes?
So, for the secretary, I kind of got confused. Would glycosylation also be mechanism, regulatory?
Okay, so I just got a question. What was for the secretory pathway? Would glycosylation be a regulatory mechanism? Glycosylation is a regulatory mechanism in terms of protein folding and quality control. But you have to remember this question's about the lysosome. It's getting to the lysosome. So you want to go directly to the point, which is what regulates whether or not a protein gets to the lysosome from the secretory pathway. Yeah. So it is a form of modification of sugars, but you want to get more specific. Okay?
Now, the other question, for the endocytic pathway, for the degradated pathway, phagocytosis and
That's how
autophagy,
yeah.
are those also examples of endocytosis? Or
Phagocytosis and autophagy are 2 methods of entering the degradative pathway to lysosomal degradation. Phagocytosis is a form of endocytosis, but autophagy is not. So there was one slide. So the question is about where do phagocytosis and autophagy fit into endocytosis and the endocytic pathway, right? And so there was, when I was talking about degradation via the lysosome, we talked about different ways that a protein might get to the lysosome to be degraded. And that was kind of in that there are these different ways. Endocytosis, receptor-mediated endocytosis is what we focus on. Phagocytosis is a form of endocytosis because it's bringing something from the outside in. Autophagy is totally different because this is degradation of something from within the cell.
but all of them end up in the lysosome.
All of them end up being degraded in the lysosome. Yes? Melanocyte, lysosomal secretion. So those are after you get to the lysosome. So those are kind of like exceptions to degradation. So that's definitely don't focus on that as being a step. That's more of like an exception of sometimes not everything gets degraded in the lysosomes, and there are other steps afterwards, and those were examples. Okay, let's move on. We've got stuff to do. This is what we've done so far. Does it seem like a lot? Yeah, I got it. So this is what you guys should be focusing on as you're studying for the exam. Last time we were kind of working through the secretory pathway, diversion to the lysosome, endocytosis, receptor-mediated endocytosis. And then my computer died on us and I sent you all away. So let's move forward. Okay, so we were talking about receptor-mediated endocytosis. And we were talking about the LDL receptor. And it's an example, remember in that paper that we did with the ricin paper. Was it the ricin paper? No. It was the HIV paper with NAP, the endocytosis paper. And there was where they were looking at recycling endosomes, and they had to draw out recycling and endocytosis and all that. So this is all related to that, right? So sometimes proteins, receptors will be endocytosed, and recycle back to the plasma membrane. And so that's what you see here with the LDL receptor. LDL is going to bind to the receptor. You'll get endocytosis. And then in the early endosome, you'll get sorting of the receptor into recycling endosomes that will then go back up to the plasma membrane. So the receptor ends up back on the plasma membrane. But the early endosome will still mature and then fuse with the lysosome. And you'll still get degradation of your LDL because you need that LDL. You need it to be broken down into cholesterol and all the parts of the whole purpose of bringing LDL into the cells so you can utilize the LDL, right? So just for fun, LDL receptor makes a round trip every 10 minutes regardless of LDL binding. So you do not actually need to have the ligand bound to the receptor for endocytosis to happen. In the case of LDL receptor, it just is constantly endocytosis, recycling. That's just how it works. So the point to really take away from this is you want to realize that there are different fates for these endosomes, right? You can have fusion with the lysosome and degradation, and then you can have recycling. And in the case of the LDL receptor, you have the receptor recycling back to the plasma membrane. So the other example is the transferrin receptor. And so this is an example of recycling of the receptor and the ligand. So with the LDL receptor, only the receptor recycles in the membrane. With the transferrin receptor, what's happening is you're getting endocytosis of your ferrotransferrin. So that's your transferrin that's bound to iron. This is a mechanism for bringing iron into the cell. So transferrin binds to iron, so it's ferrotransferrin, binds to the receptor, you get endocytosis. And as you get to your late endosome and the pH is going down, you're going to get dissociation of your iron from your transferrin. That's going to result in apotransferrin. Don't get bogged down with names, just think iron bound, not bound, right? And then the apotransferrin plus receptor is going to get recycled to the cell surface where it can sit there and bind iron again. So why do this? Why not just make more receptors and make more transferrin? Why would the cell choose to bring it in and recycle it back out? What's the advantage?
Cheaper.
It's cheaper. Exactly. It's cheaper for the cell. Less cost in terms of resources. Less time spent. Yep. Less chance for errors. I guess every time you have to go through transcription and translation, there is a chance for error to happen. Is that what you're thinking? Maybe. That seems logical. Yep. Better control. So expand on that. Where's your brain going? So by just having one cycle where you're bringing it in and going back up, you can control it more versus having like a signal transduction pathway that has to stimulate transcription translation, maybe. So I want you to be, when you're studying this, someone came to my office the other day and was like, there are all these examples you're giving me and I'm having a hard time figuring out where they go. And so these are all examples of receptor-mediated endocytosis. And I'm giving you an example of, in this case, of recycling and different types of ways things can be recycled. You can have just recycling of your receptor, or you can have recycling of your receptor and a ligand that changes depending on whether or not it's bound to iron. So just kind of keep that in mind. And the other thing to keep in mind is notice the effect of the pH in both cases on dissociation of your receptor and your ligand. As your pH goes down, you get dissociation of your receptor and your ligand, or in this case, your iron and your transferrin, and then you get it going back up to the cell surface. Okay? So why would you want to, so So we just talked about recycling, and in that case, your receptor is still ending back on the cell surface. But you can also get receptor downregulation through endocytosis. So what does that mean? And why would the cell want to send the receptor to the lysosome? What is receptor downregulation? What's the outcome for the cell?
If you already have enough of whatever ligand you need, you don't need any more. You don't need the receptor to keep bringing more in, so you degrade some of the receptors so that lots of it's coming in.
Okay, so if you're thinking about receptors in terms of bringing things into the cell, and you don't need any more of that thing, then one way to slow down it coming into the cell is to send some of your receptors to be degraded. So there's less of them on the cell surface. What else do receptors participate in? Yeah.
Well, you can bring in the ligand just to initiate the signal cascade, and then we're done with the ligand.
Okay, so I want to take a minute to talk about receptors for a minute. There are multiple reasons for receptors existing on the cell surface. One is what we think about the most, I think, up until this class possibly, which is to bind to a ligand and bring it into the cell, right? Remember when we did the paper, we talked about NAP and CD4, and I was like, well, CD4 doesn't bind to a ligand and bring it in. Its purpose is to signal and communicate with other cells, right? So there's also receptors that might bind to a ligand for the purpose of signaling. Nothing's ever brought into the cell. It's about signaling that results in gene expression. And so in that case, if signaling, so let's say the EGF receptor that binds to a growth factor, if you don't need growth to be happening anymore, you want that EGF receptor to go away so it won't bind to EGF and won't participate in signaling and cell growth. Do we always want our cells growing? No. What happens when our cells are always growing? Cancer. Oh, you guys are so well trained. I saw a hand up there. I saw a hand up over here. No? If the receptor is broken and it doesn't work anymore and there's no need for it, you should degrade it. Well, definitely if there's no need for it, you should degrade it, such as you don't want signaling to happen. But you're making me think for a minute, and I'm going to take you in a different direction. If the receptor is broken, where should it be caught before it should it ever make it to the cell surface?
Should make that out of the ER.
It shouldn't make it out of the ER, right? Because where did that receptor come from? The secretory pathway, and we've got this ERAD and all this quality control. So it should hopefully, fingers crossed, never make it to the cell surface. Because that could be bad news if it's broken, because then you could end up with cancer. Yeah. Yes.
What if it's just a protein that's kind of old, I guess, or it's been used a lot and it just needs to be recycled? It's like a new
So an old protein, right? So every protein has a half-life. Proteins don't live forever. They do get degraded on a regular basis. So one thing would be, so we talked about how CD4 is constantly being endocytosed, and sometimes it's being recycled, and sometimes it's going to the lysosome. So there is this regular turnover of proteins as well. Any other thoughts people have? This is exciting. Okay. Good. So yeah, that's the concept. So here it's showing a receptor, and what you're going to realize is this is a GPCR, G protein coupled receptor, which we will be learning about soon. And there's a ligand binding to it, right? But you can also get endocytosis, and sometimes you might get recycling, but sometimes you're going to get sent, the receptor will get sent to the lysosome to be degraded. So receptors can also be downregulated through sequestration in multivesicular bodies. That's a mouthful. What's sequester mean? Well, we've all had to deal with sequestration, haven't we? In the budget for our country. Okay, so you guys are not worried about NIH funding like I am, I guess. You guys should be worried about NIH funding because whether or not you get into graduate school depends on. Yeah, so anyways, receptors. Okay, what does sequester mean? Let's go away from government and finance for a minute. What does sequester mean? If I get to sequester you away from everyone, where have I put you? Away. In the basement, in a closet, in my office. I don't know. Somewhere where I'll probably go to jail. So sequestration, right? To hide, to keep away, right? So sometimes we get so overwhelmed with big words we forget to think about what they mean. So receptors can also be down-regulated through sequestration in multivesicular bodies. Multivesicular, multiple vesicles, bodies. So what this means is sometimes, and especially with receptors involved in signaling, you're going to get down-regulation through endocytosis. Notice there's a monoubiquitin here, regulating endocytosis. And then you're going to get, watch this. These receptors are butting into the vesicle, into the endosome, right? And so there are these multiple vesicles in this endosome. So it's called a multivesicular body. And then it ends up fusing with the lysosome and they're all degraded. So what's the purpose? Why are we doing that versus just endocytosis and going to the lysosome? What's the difference there? I believe this was on the quiz. Yeah. Well, you're going to the lysosome. Yes, you might be able to use it later on, I guess, if you were to recycle somehow. Always. But in this case, we're going to the lysosome. So why would we do this versus just plain old endocytosis? Notice, compare this endosome with the receptor on the cell surface. Notice the cytoplasmic tail, which is the part of a receptor that's involved in signaling. Notice it's in the cytoplasm, right? Notice it's still in the cytoplasm. What's to stop it from signaling when it's in the endosome, right? So by butting into the endosome to make a multivesicular body, the whole thing is sequestered away from all the cytoplasmic proteins that participate in the signaling pathway. This will become more apparent as we move towards signaling, I guess, in the next week or so. Oh my God, is that next week? Yes. No, I don't explain anything. Yeah, so compare this. So see, do you guys see my arrow? See that cytoplasmic tail? See how it's in the cytosol? So what happens with signaling? You guys have all learned very generally about signaling, and it's all about kinases phosphorylating things and sending a message to the nucleus and affecting gene expression or cell movement or something like that. But basically, it's about sending a message from the outside of the cell to the inside of the cell. And that all happens through that cytoplasmic tail, right? So if, in this case, your cytoplasmic tail has been modified into signaling, and then we decide, okay, we want to downregulate it, we're going to ubiquinate it, endocytose it. But in this case, you still have that cytoplasmic tail available in the cytoplasm, right? And so it can still be signaling. No one says it has to be on the plasma membrane, right? Once it's been activated through a ligand binding, oh look, ligand bound. Once it's been activated and the appropriate conformational changes have taken place, what's to say that it can't signal in the endosome, right? But if the whole purpose of receptor downregulation is to turn off signaling, you want to make sure that cytoplasmic tail is gone, so it can't interact with all the signaling molecules in the cytosol. So that's why it will bud into this multivesicular body, and now it's completely inside a membrane and can't interact with cytoplasmic proteins. That's that.
So the multivesicular body is like two vesicles fusing.
No, it's a vesicle butting into itself. So it's an endosome butting into itself. So just see here, it's butting in. See that butting in? Here it is. Here it's butting in, and now it's inside. So we're going to actually draw it out in recitation. Not today, but someday. Maybe next week or the week after or something like that. Okay, sit on it, think about it. Yes? So are you wondering if that's a general rule with receptor-mediated endocytosis? So when you have to think to yourself, what's the purpose of endocytosis of the receptor, right? So sometimes it's really about bringing something into the cell, like transferrin receptor and LDL receptor. In that case, MBB is not going to help, right? Because you want to recycle that receptor back to the plasma membrane. So there it's not going to happen. I believe that there are probably times when you get so if the question is, does downregulation always have a step of going into a multivesicular body? My answer would be probably not every single time, but definitely when you want to turn off signaling, it would be advantageous for that receptor to butt into a multivesicular body. Does that make sense? Okay. So endosomes can also store membrane proteins for use when they're needed. So this is showing, so here you have a glucose transporter. And they kind of just hang out in these recycling endosomes. And the purpose of those specific recycling endosomes is to have glucose transporters in them. And they're just there for when needed. And when, so normally glucose transporters, what do? They bring glucose into the cell, right? So when you have insulin binding to the insulin receptor, that's going to result in signaling. That's going to result in recycling a glucose transporter up to the plasma membrane so it can increase the uptake of glucose into the cell. So that's just one way, one example of how endosomes could be used. What kinds of cells do you think might have large intracellular pools of these endosomes containing glucose transporters? What cells need to take in lots of glucose? I heard pancreas, but pancreas secretes insulin. But what kinds of cells are going to be taking up a lot of glucose? Neurons? Muscle? Fat cells? We don't want to think about fat cells. Let's not think about them. I'm going to change my research to eradicating fat cells. No, those are they're really important. Okay, so let's talk about exocytosis or secretion. So secretion, so we've been talking about endocytosis, so things coming into the cell, but let's not forget our secretory pathway where we have things going to the ER, the Golgi, plasma membrane, out of the cell or the lysosome, right? So out of the cell is secretion. And so you're going to go from the Golgi to either straight out of the cell in an unregulated secretion called constitutive secretion, or you're going to go into secretory vesicle. And you're going to wait until you have regulated secretion. So some kind of signaling is going to result in regulated secretion. You can give me an example of something that you think might be secreted in a regulated way. What makes you sneeze in allergy season? Histamine, or pollen, correctly. Histamine, right? So there's a cool picture in the textbook of that. So constitutive versus regulated secretion. Remember ER, Golgi? Some stuff is just going to move towards this constitutive secretory pathway. No regulation required. This is how things like membrane and membrane proteins get up to the cell, right? Up to the cell surface. And then sometimes you need things that are regulated secretions, such as histamine, right? If mast cells were constitutively secreting histamine, how would we be feeling right now? would we feel good if there was no regulation of histamine secretion? It was always being made and secreted? There's like no, I got like 2 laughs. Makes me very, very nervous. He's like, I don't know, why would it be a bad thing if histamine was? Why? Who loves histamine? I took my kids, my daughter had a horrible reaction to, I don't know, it was probably just a viral rash, but when she was little, like when you have your first child, you're like, oh my God, about everything. And so she broke out in a horrible rash after I gave her yogurt. And so I'm like, oh my God, maybe she has an allergy. So we went to go see an allergist who did the skin test. And she was not allergic to anything, but like had this huge welt where with a positive control, which was histamine. So I was like, oh, she had a huge reaction to histamine. So I'm like walking around, like laughing to myself, like as I tell people about how she had such a huge reaction to histamine. But then I realized none of my friends were scientists. They had no idea what I was talking about. I was just a crazy person once again. So anyways, constitutive secretion. Stuff like membrane. Remember, your cell membrane is constantly coming in with endocytosis, and membrane needs to get sent back up with exocytosis. So that's through the constitutive pathway. And then regulated secretory pathway would be, one example would be secretion of histamine. And so the constitutive secretory pathway is your default. Remember, I keep saying secretion is your default unless you're diverted, right? So you could get diverted to the lysosome with mannose phosphate. You can get diverted to secretory vesicles, which has a specific signal involved. Or otherwise, you go out of the cell. If you're an integral membrane protein, then you stay in the plasma membrane. And if you're a soluble protein, then you just get secreted out. So there's one thing that I found to be annoying with this figure, because they're like, oh, you go ER Golgi and you can go all these ways out. And I felt like there was one other diversion that's missing from this figure. What's another way a protein could be diverted and not be secreted out, but also not go to a secretory vesicle and not go to the lysosome? Yep. Right, when it goes back to the ER, and what regulates that? What's that? Beta, right, exactly. So I felt like I should call Bruce Alberts and tell him he made a mistake. And then I realized he's just a big name, so no, he's not going to care. Okay. So specialized secretion. So when you've got regulated secretion happening, these proteins are going into secretory vesicles, and they're only secreted in response to some sort of signal. So you can imagine there's probably some sort of receptor and some sort of signaling pathway that then says, hey, vesicle, fuse with the plasma membrane and secrete, right? So where is that signal coming from? Some kind of receptor. So this is showing the release of insulin from a secretory vesicle of a pancreatic cell. And so here's your secretory vesicle, and this black stuff is insulin. Well, no, it's really black. That's just the EM. And so what you have is exactly the same concept that we've been talking about through this whole secretory pathway, which is you're going to get docking and fusion and then release of the content. So docking, you have to be thinking about docking, that must have something to do with RAB. Fusion must have something to do with snares. So it all applies, everything we've learned. And so these secretory products are actually concentrated in these vesicles. So as they're leaving the Golgi, they're going to concentrate in these secretory vesicles by basically removal of excess membrane. And also they're processed in these secretory vesicles. So have you guys heard of proproteins? Right, some proteins are translated as proproteins, meaning they're longer and need to be processed in some way. They're larger. Some sort of processing or cleavage needs to happen in order for them to become active. And so proproteins are often matured in secretory vesicles. So an example is some sorts of cytokines are not mature until they make it to the secretory vesicles and are cleaved. And this is because you don't want them to be active until it's time for them to be active. So the secretory vesicles travel to the plasma membrane and wait for the signal before fusion is complete. So they're actually up at the plasma membrane, and the snares are partially wound. And it's not until you get the signal from some signaling process through a receptor and signal transduction that you get fusion between your vesicle and your membrane, and then release of the contents out of the cell. That's why it's regulated. So this is I always love this picture. So this is a mast cell. These are your cells in your immune system that cause you to be allergic. And they are full of secretory vesicles that contain histamine. All of these round dots are full of histamine. And when you get stimulation of your mast cell, then boom, secretion. And you can see all of these vesicles are fusing with the plasma membrane and releasing their contents out of the cell and causing you to need a Claritin. Or more, depending on your level of allergy. So there's this give and take with the membrane, right? There's endocytosis. Think about it. Every single time you have endocytosis, you lose some of your membrane off the cell surface, right? So without that exocytosis happening, also, you are going to shrink and shrink and shrink. Or if you only have secretion of proteins constantly out of the cell, you're going to grow and grow and grow. So it's that endocytosis, exocytosis that are maintaining our membrane size. But sometimes we don't want to maintain our membrane size. Sometimes we want our membrane to increase, right? Do we ever want our cell size to increase? When? Q1? Right, before mitosis, before cell division. Right? Sure? What happens if you have a cell that's always the same size and you keep dividing? Then it goes to nothing. Right? So you have to think about it. If you're about to become two cells, then you should get large enough so that when you split into your two cells of the appropriate size. So the other thing to think about with secretion is with polarized cells. These are cells that have a top and a side and a bottom. Sometimes you might have secretion. You might have endocytosis of something and then secretion on another side. And there's also membrane proteins will need to be sorted to the appropriate side of the membrane. And so they'll also have specific sorting signals in their cytoplasmic cells. So these are just things to keep in mind. It's not something I'm going to spend a lot of time on, but the general theme of different sequences and different proteins help to determine where they're going to go. So that's a major theme in this unit. And that also is involved with sorting of the appropriate protein to the appropriate side of the cell with polarized cells. Okay, who's tired of the secretory pathway and endocytic pathway? I'm totally tired of it. Here you? Tired of it? So let's move on. Other compartments. Okay, quick theme alert. So you have to think to yourself, there are different ways of moving between different compartments in the cell. And I really want to take a minute to talk about this, right? You can have vesicular transport, but that's only the secretory pathway. I got an email this weekend when someone was confused and was thinking about a vesicular transport in terms of import into the mitochondria. And I definitely want to take the time right now to make sure you guys understand secretory pathway is the only pathway that uses vesicular transport. That is it. Going to the nucleus, that's different. That's gated transport. Transmembrane transport, that's using protein translocators. Remember, we already talked about one protein translocator, right? Getting into the ER. And so we'll talk about another one and getting into the mitochondria. And then, generally, what determines the correct address for any given protein? some kind of sequence, right? There's either a signal sequence that's going to dictate interaction with SRP, or there's a nuclear localization signal that will dictate interaction with an important, or there's another kind of sequence that we will talk about that forms a certain helix with certain properties that interacts with a receptor for importing to the mitochondria. It all has to do with some kind of sequence in the protein. Yes? Yeah, so endocytosis, so when I talk about the secretory pathway, I always group endocytosis into that, only because it's like this huge, you can think of endocytic and secretory as one larger kind of vesicular transport pathway. Yeah. Okay, any questions about this? Yes?
So you said that there could be like signal sequences, and then you mentioned the nuclear sequence.
Yeah, so we're going to go in great detail into it, but I just want you to realize that there are different sequences that are kind of like addresses for proteins. And some might dictate interaction with SRP, some might dictate interaction with nuclear import receptors, some might dictate interaction with mannose-6-phosphate receptors. That, you can kind of, when you start to think about protein transport in the cell, you can kind of think about that concept across the board. Okay, and we're going to go into it right now with nuclear import, right? So you need to have transport of nuclear proteins into the nucleus, and then sometimes proteins need to come out of the nucleus. So could someone think of an example of why proteins would need to enter and exit the nucleus? Yeah. So proteins that wind or unwind histones. So proteins that are involved in chromatin structure need to get into the nucleus, right? Because that's where they live and work, right? Where are they translated? Where are all proteins translated? Except, yes, they all begin their translation in the cytosol, except for ones that are going to enter the secretory pathway that then finish their translation in the ER. But yeah, all proteins pretty much start their translation or translated in the cytosol. But these proteins that are involved in maintenance of chromatin structure, that's your DNA and your histones and all of that stuff, those proteins need to be in the nucleus, right? They need to get in there somehow. Are they going to be packaged in a vesicle in the ER? No. They're not in the secretory pathway. They're going to be translated in the cytosol, and they're going to have an address on them called a nuclear localization signal that's going to dictate interaction with a protein that's going to bring them into the nucleus. What other kinds of proteins tend to go in the nucleus? And these are proteins that go in and out. Yeah. Transcription factors. Very good. Yeah, transcription factors. These are proteins that regulate gene expression. And a lot of how they work is basically, are they getting into the nucleus to bind to the promoter of whatever gene is going to be expressed? If they're not getting into the nucleus and can't bind to the promoter, you're getting no gene expression. So we'll go through a little bit of this in signal, and when we start talking about signal transduction, but most of it has to do with, is that transcription vector getting into the nucleus or not? And if it's getting in, it doesn't stay in there. It needs to go back out at some point. Will be degraded. What kinds of exiting? So we can have it in and out. Okay, so I have a quick video. I was trying to find a nice video. I know everyone loves movies. This one's okay. It's a microscopy video. Let's see if we can get it to come up without destroying. Well, now it's just like.
Nuclear import and export can be directly visualized in living cells. An
Okay, so this is showing nuclear import and export. And so this is a transcription factor that is tagged with GFP, so it's green. And so this is your nucleus, these black dots. So right now it's cytosolic, and when they start signaling through increasing calcium, what's happening is going into the nucleus. So you're actually watching the protein enter the nucleus. And so then when they take away the stimulus, it leaves the nucleus. So you're in and out of the nucleus. Transcription factor. Isn't that cool? I thought so. Okay. I kind of wanted to more of an animation, so I'm going to keep searching. Oh, you want to see it? Okay, let's watch it again. Let's watch it again. You mean you didn't get that the first time? Okay. So, why am I having a hard time now?
Nuclear import and export can be directly visualized in living cells.
Okay, so what you're seeing, these are cells, and all the black dots are nuclei. So right now, your transcription factor that's been labeled green is outside of the nucleus. But then what they're doing is they're going to add a stimulus, so it's something that's going to activate signaling, and it's going to send that transcription factor into the cell, into the nucleus. And so notice all the green goes from the cytoplasm into the nucleus. And so then what they do is they remove that signal, and then you're turning off gene expression. So you want that transcription factor out of the nucleus. And so it actually will leave the nucleus. Should we watch it once more just for kicks? If I can figure this out.
Nuclear import and export can be directly
Okay, watch where the green goes when they increase calcium. And the green is a gene regulatory protein, a protein that its job is to activate transcription. So it needs to go into the nucleus to bind to the DNA. So notice you have the green going into the nucleus.
This is done here experimentally by adding an ionophore that allows calcium to enter the cells from the needle.
And so when they take away the stimulus, it goes out of the nucleus.
And NF-AT is exported.
Isn't that cool?
Re-addition of the ionophore triggers
So just by signaling, you're inducing migration of a transcription factor into the nucleus and activating transcription. That's all signaling. And I have to apologize for those of you who aren't too familiar with signaling. We will get even more familiar with it before you know it. Uh-oh. Now I'm gonna be stuck here forever. Okay, you go away. Not you. No, you don't go away. Let's see. Oh, look at Google's thing. Okay. There we go. Okay, so that's that concept of regulated import into the nucleus, right? In that case, we're sending transcription factors into the nucleus to activate gene expression. Are genes just always being expressed whenever all the time? No, they're only expressed in response to a certain signal, right? Like, for example, if your job is to, let's say you need to fight. Let's say my favorite is always immune system related things. So let's say you need to fight some kind of infection, then signaling pathways are going to be activated that then send a transcription factor into the nucleus and activate expression of a bunch of genes that include, say, cytokines. These are signaling molecules that will then be secreted out of the cell and tell other cells, hey, we need to get ready to fight this infection. Those cytokines are not always being expressed. That would be really bad, right? Because then you would have this hyper-inflammatory, always-on immune system, which we all know is not a good thing. So, by regulating gene expression, by regulating transcription factors going into the nucleus and activating gene expression only when it's needed, that's how the cell survives. That's all of cell molecular biology right there. Well, not all of it, but that's a lot. So it's always helpful when you're thinking about these things to kind of understand the bigger picture of things, right? That's my mantra, isn't it? So when we talk about the secretory pathway, we have to start thinking about things specifically the cell, membrane proteins. When we talk about nuclear import, I want you to start thinking about proteins that work in the nucleus getting in there, and also that in and out of transcription factors, because as we start talking about signaling, it'll kind of be useful to connect those two. Okay? So there are these nuclear pores, but things don't just diffuse through them. It's a regulated import. And so this is what the nuclear pores look like, and I want you to pay attention to these kind of hairs that are sticking up, these fibrils, and they have FG repeats on them as part of the sequence, and that part interacts with your import receptors. So your nuclear import receptor is going to sense a nuclear protein through interaction with a nuclear localization signal, and then it's going to kind of hop along, like a little bunny. It's going to hop along these FG repeats and eventually make it through the pore to the into the nucleus. And there it will be released. And we'll talk about the exact mechanism. Don't worry, there's more. And so what I want you guys to realize is nuclear pore complexes. It's not like your nucleus is this static thing with 10 nuclear pore complexes. They're actually changing all the time depending upon need. Right? So if you have a lot of gene expression happening, you're going to have many more nuclear pore complexes than maybe more, what's the word? What's that word? Senescent cell. The cell that's not doing very much is going to have less going on, less gene expression, less RNA that needs to come out of the nucleus. Right? Less nuclear pore complexes. So we need this active transport. So only small molecules can actually diffuse through the nuclear pore. Those are things that are like 5 kilodaltons in size or 9 nanometers or less in size. But just for example, DNA and RNA polymerase subunits, they're pretty huge. They're 100 to 200 kilodaltons. Do DNA and RNA polymerase need to get into the nucleus? Yes, right? So that's where they work. Right? They don't just exist there. They need to be made, right? Transcription, translation, all that. That translation is happening in the cytoplasm, and then those proteins need to be imported into the nucleus so they can do their job. Ribosomes made in the nucleus need to be exported out in the cytoplasm, right? Those are about 30 nanometers in size. Way too big to just diffuse through the nuclear pore. So there's active transport that allows them to go back and forth, to move to where they need to go. Why else do you think active transport is an important or is desirable compared to diffusion?
You need a gradient when you have diffusion, active transport.
When you have diffusion, you need a gradient. Okay. Regulated, that's exactly what I was looking for. Regulated. It's regulated. If you have just diffusion, are you going to be able to activate gene expression in a very specific way? No. So by being able to control this and have this active transport, that's a form of regulation. So you can control transcription factors, for example, going into the nucleus to activate gene expression when you're supposed to. So we've been talking about how all these different proteins have different addresses that regulate where they're going to go. So for nuclear proteins, they have an NLS, a nuclear localization signal. There's also a nuclear export signal that allows proteins to exit the nucleus. So this is just showing an NLS, which tends to have lots of lysines and arginines. If you even just mutate one amino acid, you lose that import. And so this is showing microscopy where when you have an NLS, intact NLS, where is that protein? What does that look like? Does that look like nuclei to you? I know you have to get used to looking at these images, but basically this is just showing how it's all in the nucleus. And when you mutate one amino acid, you can no longer get nuclear import. So now it's all cytoplasmic. So with that video we just saw, that transcription factor that they had labeled green, it had an NLS. It had a nuclear localization signal. And then just like you can add KDAL to something and send it to the ear, you can add an NLS to whatever and send it to the nucleus in that same way. So what you have to be thinking now is, well, what is the NLS binding to, right? Because we know what the signal sequence binds to, we know what mannose-6-phosphate binds to. There's always something that it's binding to that helps to shuttle it to where it needs to go, right? We'll get there in a minute. So this is just showing the importance of the NLS. So this is a cross-section of your nuclear envelope. So here's nuclear envelope, and this in between spot here, those are your nuclear pores. And what they did is they took gold beads and they coated them with NLS, and then took pictures over time. And so this outside part is the cytoplasm, and the inside is your nucleus. And they're looking at where those beads are over time. And so notice at 10 minutes, the beads are all on the cytoplasmic side. And then over time, they're associating with the nuclear pore in the middle, right? More and more associating with the nuclear pore. And within about an hour, you start to see them coming out on the nuclear side. So that NLS will even take a gold particle through the nuclear pore. Right? Do we normally have gold particles in our skull? Right? So that kind of, you never know. Sure, you never know. I don't know what you guys do in your free time. I know what the kids are doing these days. Okay, so you have to ask yourself, oh, so what is the NLS binding to, right? So it's binding to a nuclear import receptor. So also the nuclear import receptor not only binds to the NLS, but also binds to these FG repeats on these nuclear fibrils. So this figure is showing how you can have different cargo proteins, but as long as they have an NLS, they'll bind to a nuclear import receptor. Okay? And then sometimes you might have an adapter protein that will kind of act as a bridge between your nuclear import receptor and your cargo protein. And then in that case, the adapter protein is going to have the NLS. But either way, nuclear localization signal binds to nuclear import receptor, interacts with the nuclear pore, and gets into the nucleus. Yeah. That's the FG repeats I pointed out right over here. These are these fibrils that are on either side of the nuclear pore, and your nuclear import receptor is going to bind to them and kind of hop on them until they get through to the other side.
Is it like a protein or is it like
It's protein. Yep. Everything's protein, except for when it's not. Right, so this is kind of showing how it's happening. So nuclear import receptors, they're binding to the NLS in the cargo protein, as well as the FG repeats that are on the nuclear pore. And so you can see here's your protein with your NLS, that's in purple. And that NLS is going to interact with your nuclear import receptor or important. So get used to important and nuclear import receptor, those are the same thing. You might see them used interchangeably. Nuclear import receptor is going to bind to the NLS. Then it's going to interact with those FG repeats as it makes its way to the nuclear pore. And then on the other side, it will be released. Now this is all regulated by RAM, which is a GTP binding protein. There's another GTP binding protein. But we're going to stop today for right here, and we'll finish talking about this on Thursday.
Yeah.